Good morning, everyone. I'm welcome you all to this third day of online coaching. Today topic is cell signaling. Yesterday we discussed about the cell communication. Following to the cell communication topic, we are going to discuss about cell signaling. It's an major part of Unit 4 in CSIR NET examination. Participants, please tell, is it audible? Am I audible? Okay, thank you for the response. Now I am entering to the our topic. Okay, every cells are communicated with the signals, right? As per the signals, now we are in this unit 4 part B. We are going to discuss cell signaling hormones. Which hormones are responsible? That hormones are binds with which receptors is the first topic. In that receptor, that having the different types. One is cell surface receptor, another one is cytoplasmic receptor. Among the two type, cell surface receptor is a major important one. So I am going to discuss about the cell surface receptor. Then the signaling through the G coupled receptor. G protein coupled receptor is a one important receptor present on every cell surface. It having it's a more than 500, more than 200 types are there in G protein coupled receptor. Each and every receptor having different function. They are playing a major role in the cell signaling and cell gene transcription. Then is signal transduction pathway. Then another is the secondary messenger pathway. Secondary messengers involve all at the end of the G coupled protein receptor. Many secondary messengers are activated and they are playing a major role in cell signaling pathway. That secondary messengers and G coupled receptors are regulating some signaling pathway. These are all things we are going to discuss in this class. I am lefting these three topics for the next day because these are totally deviated from our cell signaling process. So I am left that three topic. Okay, overview of this cell signaling pathway. This cell signaling pathway having n number of steps in between the activation. Okay, why we need to study the step by step process? First, the primary transduction happen in the cell surface. At the end of the session is a gene transcription. In between the signal receiving at the cell surface and the gene transcription, they having n number of steps like relay, transduction and amplify, integrate, spread, anchoring and modulate. Each and every step, now numerous number of proteins are present in the cell cytoplasm and also cell nucleus. They are asking question like ordering the process of cell signaling, arranging the cell signal transduction through nucleus to cell membrane or cell membrane to nucleus in a descending or order or maybe ascending order. It's one kind of question asked in the old CSIR question. Okay, how to arrange that? First, if they asked the nucleus to cell surface to nucleus. First step is a receiving of cell signal from the extracellular molecule. The another step is a relay. It's not, if it is not clear, no problem. I will send the PPT to your mail ID, respective mail ID. Okay. It's not clear, please understand my words. I will send the PPT to your mail ID. Okay. Cascade of cell signaling event, they are having three different proteins. One is a receptor and signal transducers, another is effector. Okay, what is receptor? It's a present on the cell surface or the cytoplasm, receiver of particular signaling. After receiving, it want to transfer to particular another protein, that is the transducer signaling. If the signal is received by some other protein, that protein will get changed itself, that is the effector protein. In between, these are the three different types. In between, they having different steps, right? Already I'm told, relay, integration and distribution. Each and every step they are need the transduction, because these steps are in between the signal receiving and gene transcription, so they need the transducers. Are the transferring molecule messengers. Okay, how this kind of signalings are transferred in 5 different types. It's a major based on this they are asking me some kind of questions, so only I am give the most important to this particular topic. Endocrine signaling is first receiving the one endocrine cell gives the signal to cell surface receptor, that gives a hormone to cell surface receptor. That receptor are getting the signal and transfer the signal through the bloodstream. It's so only it's called as endocrine, and reach the target cell. It's endocrine process. Next one is paracrine process. This paracrine process is signaling is direct cell to cell interaction. This direct cell to cell interaction happens via local mediators. This local mediators may be the hormone, steroids, this kind of don't it's just an introduction, because many of the students are here, they are can't able to pick up the questions, so only I am discussing this kind of introductions. After discussion of these questions, I will explain with some example, so please follow this. Synaptic signaling. In the synaptic signaling, some neuron cells, it is a neuron cell. This neuron cells having an axon end for receiving the signal. This axons are having the signal. That signal pass to the target cell. The target cell surface having the receptor. These are the neurotransmitters. These neurotransmitters are expressing the best way of signal for cell activation, regeneration, this kind of process. Now, another one important thing is autocrine process. After the autocrine process, autocrine is a cell to cell direct interaction. Then signaling by the plasma membrane attachment. It's a major thing. It happened in many viral infection. Signaling by plasma membrane attachment is happened that are happen for any bacterial infection to human cell. Okay, how it is happen? It's a last image. These cells are receiving the signal from another cell and producing some signaling hormone inside the cytoplasm and release to another cell. It's a cyclic process. One cell receiving signal and releases some signal for another cell. It's a cyclic process. Okay, now the major topic is hormonal hormones, signal hormones and receptors, right? I'm going to give some examples for hormonal receptors, hormonal hormones, signaling hormones. These signaling hormones are divided into 4 different groups. Okay, what are the 4 different groups? Hormonal classes are amine hormone, peptide hormone, and then protein hormone, steroid hormone. These are the 4 different types. In this 4 different type, they have different sections, but these are all the major hormones. Amine hormone having the amino acid modified groups. Amino acids having the R group, right? The modification of particular R group or carboxylic group is called as the amine hormone. Normal amino acids, they are changed at the carboxylic group or their R group. For example, here norepinephrine, it's an amine hormone. It's a modified form of the amino acid. The carboxyl group of amino acid is replaced with the benzene ring. It's one example. Another one is peptide hormone. Peptide hormone example is oxytocin. This oxytocin is 9 amino acid short chain. They are providing the question like they are providing a different type of peptide hormones, asking the count of the amino acids or asking the major amino acid present in particular peptide hormones. I will send these some short notes. Oxytocin having the 9 amino acids in their structure. In specifically it having the cysteine in a larger quantity. It's a 2 count, 2 more is there. It's a short chain peptide hormone. Okay, next is a protein hormone. Protein hormones are playing a major role in the cell transcription. Cell transcription process is fully depends on the protein signaling. Okay, protein signaling is caused by the protein growth hormones. It's just example for a protein hormone. Another one is steroid hormone. So what is steroid hormones means? It's produced from the cholesterol. We all know testosterone and progesterone. These 2 are the steroid hormones. For inhalation problem, many of them providing the steroid hormones, and then PCOD problems, they are providing the steroid hormones. The internal cell related cell related disease, they are provided the steroid hormones because that steroid drugs only providing immediate relief for the particular disease. If you want that steroid hormones passing the immediate signal, so only they are providing the steroidal drugs. Okay, in the steroidal drugs, in the hormonal drugs, they are having 2 types. One is water soluble hormone, another one is lipid soluble hormones. Water soluble hormones, why the process of this hormones are different? water soluble having different pathway and different activity, lipid hormone having different activity. Okay, first the water soluble hormone enter into bloodstream, that is not soluble, not soluble in water, so that hormones are absorbed by the cell membrane receptor, that is G protein receptor. This G protein receptor get activated by the attachment of particular hormone. Activation of G protein makes adenyl cyclase activity. Attachment of hormone and G protein makes the activation of adenyl cyclase. Why I am stressing this means it's an one major question in previous year question, because there are different type of G proteins are there. All type of proteins having different activity. If they receive a water soluble hormone means they are activating the adenyl cyclase. If they are receiving the lipid soluble receptor means that is activating that producing and activating different proteins. So only I am stressing this. Attachment of hormone to G protein, water soluble hormone to G protein receptor activate the adenyl cyclase. That adenyl cyclase activate converting the ATP into CAMP. This is CAMP is and this next pathway is the secondary messenger pathway. After conversion of to CAMP is a secondary messenger pathway. Why we are calling this as a secondary messenger pathway? Secondary messengers are small molecule. These small molecules are activating the particular protein subunit. That subunit make conversion of the energy molecules. Okay, CAMP activates the protein kinase. As protein kinase phosphorylate the protein in cytoplasm. In the activity of this protein to alert the cell activity. Cell activity fully depends on. So the water soluble molecule not enter into nucleus. It's a major thing. Water soluble hormone it's not able enter into nucleus. It's the outcome of this flow chart. Please understand the step by step process. They are provided the different steps you need to rearrange. Every part C questions are they are give the whole process. You need to arrange it. It's a major thing. Please understand the step by step process. First is once again I am repeat it. Hormone and G protein step. Activation of adenyl cyclase second step. ATP to CAMP conversion is fourth step. CAMP activity activates the protein kinase is the fifth step. Then only the action happen in the cytoplasm. They will ask the another aspect. It happen in cytoplasm or nucleus. The water soluble hormone and G protein receptor signaling happen in the cytoplasm. Okay, next thing is water soluble hormones. Lipid soluble hormones are not receiving the signal at the cell surface receptor. Why it not receiving the cell surface receptor? It's a question. Lipid molecule that are only cell surface receptor, but if in this process lipid soluble hormone that is easily attached and the lipid molecule angle automatically enter into the cytoplasm. So only here no cell surface receptor. This lipid soluble hormones are diffused through the only plasma membrane. Then hormone bind to the cytoplasmic hormonal receptor. Are trigger the gene transcription directly enter into the nucleus membrane. It's an hormone only can enter into the nucleus membrane. After entering the nucleus membrane, it transcribes the mRNA into protein molecule. This after the it's a translation process happening in the cytoplasm. Okay, these are the steps and questions are there in kind of the questions they asked. Okay, the next part of the hormonal receptor, feedback activity. The major thing in the feedback activity, I'm going to tell this now. Okay, with the example, first it's a imbalancing of glucocorticosteroid. Why we need the glucocorticosteroid? It's a major compound for our blood. That the blood pressure and cholesterol degradation property. So if it is in low concentration, the sensors in the our nerves signal to human brain. Okay, in our blood there was some it directly passing the signal. There was the lower concentration of blood concentration in our body. That signals are absorbed by the neurotransmitter. That neurotransmitters are passing signal to hypothalamus. Hypothalamus releasing the corticotropin releasing hormone. These hormones are human blood nerve system having the signal sensor, right? That sensors are sending the negative feedback. Okay, we reach the higher concentration, please stop the production of the particular hormone. Hypothalamus release that corticotropin releasing hormone. So hypothalamus stops the corticotropin releasing hormone. The feedback inhibition, the step by first, it's a major concept in particular signaling pathway. It's one example is the hypothalamus release of corticotropin releasing hormone and glucocorticosteroid. Example. Okay. This based on the location we are divided the hormonal receptors. Now till now we are discussed the signals. What are the signals? The hormones are in the cell signaling pathway. It's an hormones present in cell signaling pathway. Now we are going to discuss about the hormonal receptors. Hormonal receptors are intracellular and cell surfaces. Earlier I am told that in that we have a different type. Intracellular we have a steroid hormones and thyroid hormones. Okay, another thing is a plasma cell surface membrane plasma membrane receptors. The steroid receptors having the glucocorticoids, the mineralocorticosteroids, androgen, estrogen, progesterone. These are all the steroid hormones. Another thing, thyroid hormones, it's a T3 receptors, hormonal receptors, T3 receptors, T4 receptor. Okay, sir. It's a FSH receptor, insulin receptor, growth hormone receptor, and oxytocin receptor. In n number of receptor in the cell surface, that is differ from cell to cell. Epithelial cell having different receptor, and our macrophages having different cells are having different receptors. Just comparison table. It's in a major thing in this is synthesis and the receptors. Synthesis as a prohormone. Prohormone means it's an previous step of the signaling. So peptides are synthesized as a prohormones. Steroids are synthesized from the cholesterol. Amino acids are derivatives of leucine. It's a major three thing. Then solubility is vary. The receptors of particular peptide hormone is on cell surface. Steroid hormone, receptor for steroid hormone is present in the intracellular receptors. The amino acid derivatives of the receptor are present at the cell membrane. Some of the thyroid hormones having the new all of the amino acids having the receptors at the cell surface. Thyroid hormones are having the receptors at the nucleus membrane. Okay, so some of the examples are here. This is also the another chance of question, because amino they will ask insulin is a what kind of peptide, what kind of hormone. Okay, peptide kind of peptide. Insulin is a peptide hormone. Glucagon, prolact, ACTH, gastrin, parathyroid hormone. These are all the peptide hormones. Steroid hormones, cortisol. Previously I am told that in previous slide, right? It's in just examples. Okay, these are all. It's a very, very important thing. I am faced with this question in the CSIR exam also. Because they are provide the hormonal name, they may ask the chemical nature, they may ask the action. So I would like to because it's a very important part. It's also confusing also, because for example, glucagon, they are stimulate the glucose synthesis. Their another insulin is stimulate the glucose uptake. In examination we will confuse. It's a major blending mistake. It's taken from one article itself. I'm included in this particular slide. Adrenaline, it's a source is the adrenal gland, increasing the blood pressure and the heart rate. The cortisol, it's also produced from the adrenal gland. It's a steroid hormone, affect the metabolism of the proteins, hormones and lipids. The yeast, it's produced from ovary. It's a steroidal hormone. It's induce and maintain the secondary female sexual characterization. Glucagon, it's produced from alpha cell of pancreas. It's a peptide hormone. Stimulate the glucose synthesis. Insulin, it's pancreas. Glucagon is alpha cell pancreas. Insulin is beta cell pancreas. It's a kind of the protein hormone. Stimulate the glucose uptake. Testosterone is a testes. It's produced from testes. It's a steroid hormone. Use and maintain the secondary male sexual characterization. Then thyroid hormone, it's produced from thyroid gland. Amino hormone, stimulate the metabolism of all cells. If they ask cortisol, means that stimulate the metabolism of protein. Cortisol stimulate the metabolism of protein. Thyroid hormone stimulate metabolism of all cell types. Okay, these are the main two difference between this. Another is epidermal growth factor receptors. It's produced from various cells. It's a protein hormone. Stimulate the epidermal and cell type proliferation. Epidermal growth factor is responsible for stimulate the proliferation. Platelet derived, it's a protein. It stimulate the cell. It's also stimulate the proliferation. And nerve growth factors, various produced from various tissue, promote the survival of certain class of neurons. It's promote the growth of their axons. Survival of certain class of neurons, it's promote the growth of the axons. Transforming growth factor, it's inhibiting the cell proliferation. Okay, now it's a task. The epidermal growth factor promoting the proliferation. transforming growth factor, it's inhibiting the cell proliferation. That it's a difference. Histamine, it produced from mast cell, it's an amino hormone. It causes blood vessel to dilate, become leaky. It causes the inflammation. Histamine causes the inflammation. Nitric acid, its nitric acid is a dissolved gas. It's a hormone, because this hormone acts, it causes the smooth muscle relax. It's regulate the nerve activity. These are all the local modulator hormones, particular human cell signaling. And the neurotransmitter, the cell signaling, right? It's a very important part. The acetylcholine, this acetylcholine is a derivative form of choline. It's a excitatory neurotransmitter at many nerve cells. The central nerve system having the and receiving the signal from the neurotransmitter acetylcholine. The demand of acetylcholine will makes many problem. So the neurotransmitter, this having the acetylcholine as a hormone, this acetylcholine hormone having the major important role in the central nervous system. That another one is gamma amino butyric acid. This is also produced from the nerve terminals, derivation of the amino acid glutamic acid. That is the glutamic acid derived hormone at inhibitory neurotransmitter. The another first one is the excitatory neurotransmitter. Now this is the inhibitory neurotransmitter. Please understand the both function. Another one is the delta. Delta membrane is a membrane protein. It inhibiting the neighboring cells. Specialization is a signal cause. Okay. It's just a schematic representation. What is a cell surface receptor? Another is intracellular receptor. Next is signaling of heart muscle cell, signaling of salivary gland, signaling of the muscle cell. First example is signaling through the heart cell. Acetylcholine attached cell surface receptor of the heart muscle cell. That heart muscle cells are forced for the contraction. It's a forced contraction. This contraction make the heart functioning. Heart functioning is fully depends on the acetylcholine signaling receptor present on the cell surface of heart muscle cell. The another thing is the salivary gland. Salivary gland is having the receptors on cell surface. These receptors are helps to secrete the internal cell compounds. Another thing is the skeletal muscle cells. Skeletal muscle cells are ion channels based thing. First is the contraction function is secretion. Another function is contraction. This muscle contraction based reaction happen in muscle cell by the channel opening and close, but in heart cell it happening of the acetylcholine receptor. In the plasma membrane we have different type of signals. Okay, it's another important thing, that cell surface receptor G protein, G coupled receptor, It may be a barrier, it may be a buffering compound, it may be the internationalized compound, it may be the plasma membrane signal receiver. These are main. There is many type of G coupled proteins are there. These G coupled protein receptors are passing the signal to the genetical transcription process. So plasma signaling, the but the signal receiving type is plasma membrane signaling. All the things are happen in the signal, the cell itself. These are all the functions I am going to explain with the image in upcoming slides. Okay. Signal transduction pathway is a G coupled protein. The topic, another next topic. First is a hormone and sorry. Signal transduction pathway. In this signal transduction pathway, you can see the inactivated G coupled protein in the primary step. Okay. Primary step means the protein alone present on that receptor alone present on the cell surface. That is a green color. Cell, the receptor. It's an upper cell membrane. That upper cell membrane, it's a receiving the signal from external factor and pass the signal to the internal cell membrane. In the internal cell membrane, we are having the G coupled protein. This G coupled protein having the alpha membrane, beta membrane and gamma membrane. This alpha membrane receiving the signal from the external receptor get activated by the attachment of GDP. This signal produce the GDP convert the GDP to GTP. After the conversion of GTP, the alpha subunit alone get activated. After the receiving of this activations divided from beta and gamma. Now at the signaling at the in between of signaling pathway, alpha subunit alone activated and move from the whole part of the G coupled protein, and they are both are having signals, both are in activated form. Okay, next is the ion channel based receptor. Ion channel based activation of G coupled protein. Ion channel based activation. What is based activation? In this ion channel based activation, the GTP bound activated alpha subunit interact with particular target protein. Target protein may be the any disease responsible, or may it responsible for any major gene transcription process. Many factors are there. Target protein is unknown. The process of ion channel pathway is binding of activated alpha unit of G protein binds with the target protein. That target protein activated the further process, the further hydrolysis process. After the hydrolysis process, the GTP converted into GDP. Okay, so this particular portion GDP is converted, right? Due to this conversion, the alpha subunit become inactivated form. First, with the GTP, it is in activated form. After binding of target protein, hydrolysis happen at the G alpha unit of G protein. So that G protein is become inactivated. Okay, why? What is the purpose of inactivation? It's this because of inactivation. So that beta and alpha subunit, the gamma subunit comes to the alpha, make a arrangement, and it become a alpha coupled receptor, a G protein coupled receptor. Now it's an ion. Once receiving the signal, it activates the target protein. After releasing of a signal, it's back to the form. Okay, next is the enzyme based activity. This based activity is activating the alpha beta complex and the GTP activity. The GTP activity are activating the target protein. The target protein messengers is on another short form of this particular portion. Okay, next to the our acetylcholine receptor based one, right? It's previously I am discussed. This is the secondary messenger based activity. Please listen, because it's a very important thing. It's ion channel and secondary messenger activity. They are asking questions based on the ion channels. It's a potassium ion channel activity. I am just give the example for this. It's a potassium channel activity. Yeah, acetylcholine is binds to the cells of its receptor, pass the signal to as usual alpha unit of G protein. This alpha unit get activated. This activated alpha subunit open the channel at the cell surface. This channel is closed, right? First step. This activation of alpha subunit opening the cell surface receptor. This cell surface receptor allow the potassium ions to release from the cytoplasm to extracellular space. Why it want to release for extra cell surface released from the cytoplasm? Okay, these potassium ions are the signals of particular cell. These signals are recognized by the opponent cell and other cells, and activating the particular programming. After releasing of particular amount, there is a limit, right? Not all the potassiums are released from this cell. Small amount of, after releasing of particular amount of potassium, proteins are G protein get inactivated. After inactivation, it's back to the form. It's a one-step process of ion channel opening process. Likewise, we have different calcium and magnesium process. This is overall activity of G coupled protein receptor. Maybe image is not clear. I am just explain the functions. It explain with 5 functions. Biogenic amines. These are all the receptors for G protein. Biogenic amines, amino acids and ions, lipids, peptides and other compounds. G proteins may receive the signals from biogenic amines, the dopamine, acetylcholine, noradrenaline, this kind of compounds. It may receive the signals from amino acids, calciums, glutamate, that kind of compounds, lipids, leukotrienes, prostaglandins, lipids. Another receptors is a peptide receptors, and proteins, cytokines, angiotensins, thrombin. Another things are, it may activated by the light, it may activated by the nucleotides, it may activated by the odorants. These are the signals for G coupled proteins. Based on the receptor activity, alpha subunit activity, we can, this alpha subunit having the 4 different types. This 4 different type activity, it will cause the different function, it will do the different function. It's a basic image. Okay, this G coupled protein receptor is divided into 2 types based on their activity: homologous desensitization and heterologous. Homologous process, first is a homologous process. In this homologous process, the signal is passed to all type of the G alpha, gamma beta, the receptor site, and then it passes the signal to GRK cells, and it's convey. These are the decline in the cardiac function. It's cause the it increasing the blood pressure and cardiac decline. The non-canonical pathway, it's a limited and reduced G coupled protein receptor signal receiving. That receptors are internalized, so there is no signal receiving in the particular G coupled protein receptor. We can control, we have the controlled heart rate. Another thing is what are the receptors are responsible for these functions. In this image they are explained. The muscarinic receptor as an adrenergic receptor. These 2 receptors are activated. These 2 receptors are activating the activated with the G coupled. This G coupled protein activate the acetylcholine compound and in the internal compounds that makes the cardiomyocytes and apoptosis, smooth muscle contraction. Smooth muscle contraction is a very it's a very dangerous thing happen in the, it want to be in particular limit. If that limit does exist or decline, we face many cardiac related problem. The adenosine receptor and adrenergic receptor, they are passing signal, then the same process also. Then serotonin receptor and angiotensin receptors are based on this for responsible for smooth muscle contraction and hypertrophy and cardiomyocyte survival. This is the secondary messenger pathway related cascades. It's receiving of signal to particular receptor that make the integration and adaptation. That signals, after getting a signal, it amplified to all cells and reach the target cell. This amplification, we need multiple number of signals, that is the secondary messengers. Okay, it's another example. I told right, potassium channels are there, magnesium ions are there. This is an example for potassium channel and magnesium channel. Growth factors receiving the potassium channel is the exporter of Mg2 plus, and particular one channel, one transmembrane receptor is expressing the Mg2 plus and Na plus, that activating the growth receptor on the cell surface. That activation of growth factors are promoting stimulate the P13 potassium, then activate the motor protein. This activation of motor protein cause the autophagy, protein synthesis, endocrine, this endoplasmic reticulum, transcription and growth of particular cell. Mg2 plus signals are responsible for ATP binding and enzyme activity. They are present and they are activating the mitochondrial that they are produced from the mitochondrial regions. The mitochondrial activation is responsible for Mg2 plus ions productions. It may cause the receptor. It may the another question is activation of the Mg2 plus channel by TPRM7 protein and CNNM3 proteins. These are all the another in the overall Mg2 plus secondary messenger is the Mg2 plus that activating different pathway. One of this growth factor pathway, the another one is mitochondrial based activity, another one is the exporter pathway. These are the next topics we are going to discuss in tomorrow class. Cell communication, the hematopoiesis, gender principle of cell communications, adhesions and neurotransmissions. Now we are going to discuss about some questions. Last day itself I sent some questions. These are the questions are not relevant, mostly relevant to our theory. It's fully relevant to the examples. Please wait 1 minute. I'm connecting to the okay. Actually I'm not directly connected to the questions. I'll discuss in the session, and also I will send the question, this today question. Every day I will send some questions. I'm also taken from the internet, already available websites, all websites. I will send the question. Yesterday I've sent the part B question of unit 4. Today I will send the part C questions of the unit 4. Okay, I wish to discuss one question. The mouse bone marrow cells were fractionated into the mouse bone marrow cells were fractionated to drive stem cells antigen. This antigen cells cultivated with interleukin 3. In this interleukin 3 are the granulocyte macrophage colony stimulating factor, or macrophage stimulation factor, or granulocyte colony stimulation factor. The question is, most now numerous various colonies were obtained the culture stimulated with. Okay, the bone marrow cells were fractionated to stem cell antigen one compound one cell. First they are fractionated to stem cell. These cells are cultivated with interleukin, granulocyte macrophage colony, stimulating factor, macrophage stimulation factor, granulocyte colony stimulation factor. The most numerous and various colonies were obtained in the interleukin 3 based grown cell. This is a question. The most numerous and varied colonies were obtained in the culture stimulated with the interleukin 3. Why? Because the interleukin 3 is a signaling compound which pass the signal to I think now we can see that. Okay. Okay. I'm just told the one question alone. Mouse bone marrow cells were fractionated to stem cells antigen. It's a statement. These stem cells are grown with interleukin 3, granulocyte macrophage colony stimulation factor, macrophage stimulation factor, granulocyte colony stimulation factor. They are grown with these four different compounds. They are grown with cultivated with interleukin 3, granulocyte macrophage colony stimulation factor, macrophage stimulation factor, granulocyte colony stimulation factor. Most numerous various colonies were obtained from which culture stimulated with. They are asking the question with which compound they are shown gives better result. The first step, they fractionated into stem cell. These stem cells are grown with four different factors, that are the four different hormones, four different signaling compounds. Among these four different signaling compounds, interleukin added culture gives the better result. Is the output. Interleukin 3 added culture gives the better result. So this is okay. Thank you all. I will send the questions as usual. I am send the questions to your Google Classroom and mail IDs. If anyone having doubt, please tell in chat box. If you have any doubt, please put in chat box. What are the type of cell signals in plant? Okay, plant signal is, now we are discussed fully about the animal cell signals. Plant signals are totally different from our human cell. This is a major I think it's a topic in tomorrow class. Yeah. Oh, sorry. Actually, that is the second. I left 3 topics, right? Bacterial and plant 2 compound system. In that area, that light signaling in plant. These are all 3 different. They are totally deviated from this human cell signaling pathway. So only I left the particular portion. Cell signaling plant is a tomorrow session. Y variant colonies. Okay, it's their interleukins are cell signaling compounds. They are the T cell based system. They are in macrophages are activated once the immune response is activity. They are not fully activated. Macrophage cells are not activated in every condition. They are activated in only the 1 minute. I'll show the question, so I'm taking this. Give a minute. I'll add the questions to PPT. I think now you get the question. Interleukins are easily transfer the signal from one cell to cell. For cell cultivation and growth, they need the communication, better communication between the cell to cell. So only they are the interleukins are playing a major role. Every cell growth, they need a communication. This communications are happen only happen in many way, but interleukins 3 are transferring the signal, effectively transfer between the cell to cell. So only we are obtain the more number of cells in the particular culture. Is the bioactive compounds from the plants can used as a drug molecule kind of amino acid derivative compound. Yeah, this is the one name Aarti Saroja ma'am. The question is the bioactive compound from plant which can used as a drug molecule is a kind of amino acid derivative compound for signaling. The plant derived compounds are signaling. They are not in the the amino acid derived compound. Okay, they are mimic the amino acid process. The many phytochemicals are having the 50% similar structure of the amino acids. So the plant compounds are activating this pathway, are blocking the signaling pathway. It's not fully play the role like amino acid, partially playing the role. What happen in any hormone? In all, Ishwari ma'am, due to the hormonals, we get the signal that is I was explained in the particular slide. Hormonal imbalance. This imbalance is a cyclic process. The imbalance is recognized by the RS sensors. These sensors are give the signal origin, that origin producing hormone, that hormone releasing the particular compound, the imbalance. That imbalance is recognized as a feedback, negative feedback. After particular level, we production of particular hormone with the cell signaling receptors. That after the end of negative feedback, there is a hemostatic process happen in the bloodstream. Yeah, interleukin 3 is signaling is it comes under the paracrine signaling. Okay, I think we will wind up this. Okay, thank you. To cell direct interaction happen in the autocrine, but the paracrine needs a compound. Paracrine needs a intermediate compounds, the molecular mediator for the cell signaling. The autocrine no need any intermediates. It's direct cell to cell membrane interaction. Thank you so much. If you have any questions, please post it in Google Classroom. If you have any you can also post it in Google Classroom. Thank you everyone. I'm going to end this streaming.